All right, 1107, chapter 14 deals all about Mendelian genetics. And most of the stuff that we're going to learn today in this chapter was figured out by a guy named Gregor Mendel. Right? This was actually an Austrian monk that lived in the 1800s and might be one of the most famous, if not the most famous biologist ever. Maybe some people have Darwin ranked above him, but Gregor Mendel, what he figured out was so huge and groundbreaking that every introductory biology textbook written in the last century has an entire chapter devoted to just what he figured out. Everybody learns about Mendel's pea plants, and you might have learned about them in your high school biology class. But we're going to go a little bit more in depth with his stuff here in this class. So basically, what he was trying to figure out, he was trying to, figure out the mechanism behind inheritance. Why do the progeny of sexual reproduction look kind of like their parents? You might have noticed you look kind of like some combination of your parents. You may have your father's ears and your mother's nose, and your grandfather's attitude. You're kind of a mishmash of all the different your parents and your parents' parents. And if you have kids, then your kids are probably going to look and act a little bit like you. So why do offspring look like their parents? And so his simplest experiment, Gregor Mendel's simplest experiment, was where he took pea plants, right? Just garden peas. And he noticed that some pea plants, they have purple flowers, and some pea plants have white flowers. And if you take a purple flower plant, breed it with another purple flower plant, and you keep doing that over and over and over again until you get nothing but purple flowering plants. So if you breed that purple flower plant with another of its siblings, you get purple flower plants. And if you do that, then what you have is what we call a true breeding purple flower plant. Now, he didn't know why. He didn't know why these had purple flowers. All he knew is that he could breed them with themselves over and over and over again and get nothing but purple flowering plants. So he knew that they were true breeding. Whatever was in there that was making purple flower plants, that's all they had. And then he did the same thing with the white flowering plants. He just kept breeding white flowering plants to white flowering plants over and over and over again until he got true breeding white flowered pea plants. So he took his true breeding purple flower pea plants and his true breeding white flower pea plants, and he crossed them together. So he took the pollen from one and pollinated the flower of another. So he took the pollen from the purple flower pea plant and used it to pollinate the flower from a white flower pea plant. And here's what he got. All purple flowers. Every single one had purple flower pea plants. And so this generation here, we'll call it, and this will come up a few times over the course of this lecture, This is what we'll call the P generation. P for parental. This is the P generation. This is the F1 generation. And if you cross these two together, then you get F2, and so on and so on and so on. So, his P generation was true breeding purple flower plants and true breeding white flower plants. When he crossed them together, he got nothing but purple flower pea plants in the F1 generation. No matter what, no matter how many times he did it. Now, then he took his F1 plants, right? He took an F1 pea plant, crossed it with another F1 pea plant. So these were purple from that F1 generation. These were also purple. Crossed them together. He got 75% purple and 25% white. Right? This generation right here, right? I alluded to it a second ago. This is the F2 generation. So when he took an F1 individual, which had purple flowers, and bred it with another F1 individual, which also had purple flowers, he got 75% purple flower pea plants and 25% white flower pea plants. So this was really his simple and maybe most famous experiment. And before we go into unpackaging why this is how it works, let's get a few definitions straight so that we can talk about it. First up, a character is just a heritable feature that can vary between individuals. In this case, he was looking at the flower color character. So this is just a heritable feature. Heritable just means you can inherit it from your parents, right? The purple flower pea plants got that purple color because they inherited that feature from their parents. It's a heritable feature that can vary among individuals. Now that flower color character can have a number of different traits, right? You can have white flowers or purple flowers. So the white flower is one trait and the purple flower is another trait for that character. So one trait for the flower color character is purple, another one is white. So these are just variants for a character. Turns out the flower color character was determined by the sequence of a gene. Mendel didn't know this. He didn't know what genes were. But for right now, we'll just think of genes as a discrete unit of hereditary information. and we'll take it a step further and say a discrete unit of hereditary information encoded in DNA. And different versions of a gene can cause different traits. So there's one gene that controls the flower color in pea plants, but one version of that gene gives purple flowers, and another version of that gene gives white flowers. And so those different versions of the gene, we call them alleles. So an allele is just an alternative version of a gene that can produce different traits. So it's an alternative version of a gene that can produce different traits. In this case, either purple flowers or white flowers. So, just now we have all this vocabulary, right? The flower color character was encoded by a gene, and that gene has two different alleles. One allele encodes for the purple flower trait, and then the other allele encodes for the white flower trait. So, erase all that. So, the gene encoding flower color in pea plants, encoding for that character, has two alleles. All right, one of them encodes purple, one encodes white. So, purple, we'll call that capital P. Right? That's how we denote the allele for purple color in the pea plant flowers. And the other one, lowercase p for white. Right? The reason that we use capital P and lowercase p instead of P and W is because it turns out that the purple allele is what we call a dominant allele. And so we'll go into what that means in a minute. Not what you think, probably. And the white flower allele is what we call recessive. So that's why we use lowercase p. So capital P for purple, lowercase p for white. So at this point in the semester, you already understand that an individual that is produced by sexual reproduction is going to receive one copy of the gene from one parent and another copy of the gene from another parent, right? Because you get one set of chromosomes from one parent and then another set of chromosomes for another parent. So you're only going to get, for some genes you'll get multiple copies, but for this particular gene, there's only one copy of that gene. You get one, or the pea plant's got one from one parent, and then the other one from another parent. So the possible combinations that you could get, right, for any one individual pea plant, there's only 3 possible combinations of these alleles that they could get. One is purple, purple. The other one is purple, white. And then the other one is white, white. Right? So these possible combinations of alleles, we call them genotypes. Right? These are your genotypes. Purple, 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 white, or white, white. If the pea plant got 2 purple flower alleles, then they make purple flowers. If a plant got a purple allele and a white allele, then they still make purple flowers. That's one of the reasons that we call this the dominant allele. Because if the individual got a dominant allele and a recessive allele, then the dominant allele is the one that gets expressed. So an individual that got one of each still has purple flowers. That individual only shows white flowers if they got 2 white alleles. So there's 2 things going on here. There's the genotype, all right? There's the actual combination of alleles. And then there's what gets expressed, which is what we call, I'll do a different color. This is what we call a phenotype. Right? So the phenotype is determined by the genotype of an individual. So this individual might get purple allele and a purple allele, so they're going to have purple flowers. This one gets purple allele and a white allele, but the purple allele is dominant, so it has purple flowers. That's its phenotype. So even though these 2 have the same phenotype, which is the outward expression of those genes, they may have different genotypes. So the genotype is just the combination of alleles in that individual, and the phenotype is an organism's observable traits. Right? This one has the white flower trait. So the phenotype is just what trait is actually expressed. I'm going to erase this part. And I'm going to give you a couple more definitions. Most of you probably already know these. This is all review so far, probably. Homozygous and heterozygous. So, an individual is homozygous if it has 2 identical alleles for a gene. And an individual is heterozygous if it has different alleles for a gene. So a heterozygous individual has two different alleles for a gene. Right? Just to clarify, these are both adjectives. So you say an individual is heterozygous or homozygous. The noun version is homozygote or heterozygote. Right? So you can say things like a homozygote is homozygous. Right? So I'm going to get those mixed up. Probably won't. Okay. So those are more definitions for you. So if you look at that individual that got two purple flower alleles, right? This individual is homozygous. It is homozygous for the dominant gene. So we say it is homozygous dominant. That implies that it's homozygous, right? So it has two identical copies, two identical alleles. But those alleles are the dominant alleles. So homozygous dominant just means it has two dominant alleles. As opposed to homozygous recessive, which means that the individual has two recessive alleles. Okay, so now that we've got all that vocab under our belt, right, I'd say hit pause and go review that vocab so that I can talk about it. And then come back. So we're going back to Gregor's garden peas. So when he took a true breeding purple plant, what he actually had, right, now that we understand inheritance, what he actually had was a homozygous dominant individual. So the genotype for that individual was purple purple. And when he had a true breeding white flower plant, what he actually had was a homozygous recessive individual. So those were the genotypes of the individuals that he was crossing in his P generation. So I don't have to write out true breeding anymore because we know what it actually was. So in his P generation, this is the cross he was doing. Purple purple crossed with white white, right? That was his P generation. So, what do you get when you take a gamete from this individual and combine it with a gamete from this individual? Well, there's only one possible combination, because the offspring are all going to get one set of chromosomes here and one set of chromosomes from here. The only possibility is that they get a purple allele from this parent and a white allele from this parent. So every single one of the offspring of this cross had the same genotype. which was purple, white. 100% of them had that genotype. That's in the F1 generation. Right? So that's why all of the offspring of this cross were purple, because all the offspring had the exact same genotype, which was purple-white. Since purple is dominant, purple is the one that gets expressed. All of these individuals had purple flowers. All right? Now, then he took multiple F1 individuals and crossed them together. So he took an F1 that had a purple-white genotype, crossed it with another F1 that had a purple-white genotype, right, in the F1 generation. Cross them together, what do you get? Well, now you've got a few more possibilities, right? One possibility is that the offspring got a purple allele from here and a purple allele from here, right? So that's one possibility. Another possibility is that the individual got a purple allele from here and a white allele from here. So purple and white. Another possibility is that the individual got a white allele from here and a white from here. All right? Those are all the possible combinations. So these offspring here in the F2 generation, these offspring were purple, these were purple, and then the recessive allele shows back up. The recessive phenotype shows back up in the F2 generation because these individuals had two white alleles, so these had white flowers. But he got a consistent ratio, right? He always saw 75, in this F2 generation, he always saw 75% purple and 25% white. So to illustrate why, we're going to use a tool that, again, you've probably seen it before in high school. It's called a Punnett square. So we'll draw a square. And what this is it's a tool for predicting the ratio of genotypes in the offspring of a cross. So in this case, we were crossing a heterozygote with another heterozygote, right? A heterozygous female with a heterozygous male. So the way to use a Punnett square is on the left side and on the top side, you put all the possible gametes. So we'll put all the possible female gametes on the left side, and we'll put all the possible male gametes on the top, right? One here, one here, one here, and one here. So think about what are all the possible gametes that could be made by this female individual, right? I'll let you pause and look back over your notes and guess, and then come back and see if you got it right. So pause. All right, you're back. What were the possible gametes that you could make? One possibility is that the gamete has a dominant allele, and the other possibility is that the gamete has a recessive allele. Those are the only two possible things that could happen. All right? Up here, the male can also produce two different gametes. One with the dominant allele, the other one with the recessive allele. So if this gamete from the male combines with this gamete from the female, then the genotype of the offspring is dominant-dominant. If this gamete combines with this gamete, then you would get dominant recessive, because this offspring gets this dominant allele and this recessive one. Generally, the convention is to put, for a heterozygous individual, you just put the dominant allele first. Doesn't matter where it came from. If it's homozygous or if it's heterozygous, you just put the dominant one first. All right, here, this one got a dominant and a recessive, so big P, little p. And then this one right here got the recessive allele from that parent and the recessive allele from this parent. So it's little p, little p. So this thing is called a Punnett square. And it's a pretty good way to visualize all the possible progeny genotypes and their expected ratio given the genotypes of the parents. And what I want to point out here is that each box here represents a possible outcome, right? This is what you get if the gamete from the female has that allele and the gamete from the male has that allele, right? That's one possible outcome. Here's another possible outcome. Here's another possible outcome. And here's another possible outcome. And because of the randomness of, independent assortment back in metaphase 1 and metaphase 2 in meiosis, each of these is equally likely to occur. There's a 50-50 chance that this allele gets passed on from the mother, right? It's like flipping a coin. Half the time the mother's going to pass on that one. The other half the time the mother's going to pass off that one. So you'll get one of these two half the time, and you'll get one of these two half the time. Same way with the gametes from the male parent. He's going to pass on that allele half the time, and he's going to pass on that allele half the time. So you'll get one of these half the time and one of these half the time. So each square has an equal probability of happening. So you'll get this result 25% of the time, this result 25% of the time, this result 25% of the time, and this result 25% of the time. So I'm going to leave that square up. So there's only 3 possible genotypes. There's 4 squares here, but there's only 3 genotypes, because you'll notice you see the same genotype twice. You see heterozygote in 2 different squares. Even though it's the same genotype, it resulted from different combinations of alleles, right? This one got the recessive from the mother and the dominant from the father. This one got the dominant from the mother and the recessive from the father. So even though it's the same genotype, there was a different sequence of events that got there. So these are 2 separate events with the same outcome. So let's look at the genotypes and their ratio. All right. What are the genotypes you could see? Homozygous dominant, heterozygous, and homozygous recessive. They happen in a ratio of 1 to 2 to 1. Right? You see 1 homozygous dominant individual right there. You see 2 heterozygous individuals here and here, and you see 1 homozygous recessive individual here. So this happens in a ratio of 1 to 2 to 1. But Gregor Mendel didn't know that. All he knew, all he was looking at was the color of the pea plants. So he was looking at the phenotypes, trying to figure out what was driving them. So he was looking at the phenotypic ratio. All right? For the phenotypic ratio, there's only two possible outcomes. Either the flower had purple plants, either the plant had purple flowers, or the plant had white flowers. So either purple or white. And what he saw was for every 3 purple flower pea plant, he would see 1 white flower pea plant. So they happened at a ratio of 3 to 1. And with the Punnett square, now you can see why. This individual is purple. This one's also purple. This one's purple. This one's white. There are 3 purple plants and 1 white plant, or 3 purple flower plants and 1 white flower plant. So that's why he saw the ratio that he saw here. All right, so here's a problem for you. How do you determine the genotype of a purple flowering plant? Let's say you're trying to reproduce what Gregor Mendel did, and you go out and buy some garden peas from the Home Depot, and you grow them up, and you see that their flowers are purple, and you want to know what their genotype is. So how would you figure that out? Right, without trying to run a PCR reaction and go into a laboratory, right? There's a way that you could do it just by breeding that plant. Because if you have a purple flower pea plant, there's 2 possibilities. One possibility is that it's homozygous dominant, right? The other possibility is that it is heterozygous. Either one will give you a purple flower pea plant. Right? And you want to know whether that individual is homozygous dominant or heterozygous. So to figure that out, you do what is called a test cross. Right? So in a test cross, what you're trying to figure out is the genotype of an unknown individual. Right? I've got an individual with purple plants. I want to know what genotype it is, so I do a test cross. So I'll take that unknown genotype individual, and cross it with the homozygous recessive, with the homozygous recessive individual. The recessive homozygote. So I would take that purple flowering pea plant and cross it with a white flowering pea plant, because we know now that all white flowering pea plants are homozygous recessive. So there's 2 possible outcomes of this test cross. Possibility 1 is that my mystery purple flower pea plant is homozygous dominant. All right, that's one possibility. Homozygous dominant. So if we do the Punnett square for that cross, right? we'll put that up here, all right? This is our mystery individual if it's homozygous dominant. Over here, we put the genotype for our recessive homozygote. So the genotype for this individual is little p, little p. So, what are the possible combinations? Right? Right here, this individual, we get a dominant allele from here and a recessive allele from here. So it's heterozygous. Dominant, recessive, this one's also heterozygous. Oh, oops, try that again. What am I doing here? There we are. All right, turns out there's only one way this can play out. Every offspring is heterozygous. Heterozygous, 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 heterozygous. So we would get 100% heterozygous offspring and 100% purple flowers, right? So that's one possibility. What's the other possibility if you have a purple flower pea plant, right? This is what happens if you're looking at a homozygous dominant individual, but a heterozygous genotype would also give you purple flowers. So the other possibility is that you're looking at a heterozygous individual. So if you do that cross, right? If you cross a heterozygous individual with a recessive homozygote, which is little p, then you get this outcome here. Right? So here, there's two possible offspring that can be produced. Right? There's heterozygous, and then there's homozygous recessive. And each one has an equal probability of happening. So you see these at a 1-to-1 ratio, right? So the genotype happens at a 1-to-1 ratio. And the genotypes are heterozygous and homozygous recessive. All right, that's our genotypic ratio. The phenotypic ratio is also 1-to-1. Purple to white. So, if we go back to, bear in mind what it is that a test cross is, what you're trying to figure out with the test cross, right? The test cross lets you determine the genotype of an individual by crossing that individual with a recessive homozygote. So if I have a purple flower pea plant, it could be heterozygous or it could be homozygous dominant. So if you cross it with, if it's homozygous dominant and you cross it with a white flower pea plant, then you would get 100% purple flowers. But if it's heterozygous and you cross it with a white flowered pea plant, then the offspring will be half purple and half white, because half of them will have a heterozygous genotype and the other half will have a homozygous recessive genotype. All right. So let's see. That cross that we've been looking at, right, where we're looking at a cross between two individuals that are heterozygous for one character. This is called a monohybrid cross, right? Because these individuals are heterozygous for one gene. Right? We're only looking at one gene, mono. So let's draw that Punnett square again. Just so I can put it up on the board and talk about it. Big P, little p. Big P, little p. Oops. All right, so we're looking at our Punnett square again. This was for a heterozygote crossed with another heterozygote. Here's a question for you. What are the chances of getting, remember these are the females and these are the males. What are the chances of getting this allele from the female parent and this allele from the male parent? Right? That'd be a good one to write down. What are the chances of receiving a dominant allele from the female parent and a dominant allele from the male parent? You figure that out by multiplying the probability of this happening by the probability of this happening, right? So the female is going to pass along this allele 50% of the time, half the time, and the male is going to pass on this allele half the time. So the answer is 1/2 times 1/2, right? 1/2 multiplied by 1/2 is 1/4, and it just so happens that this square takes up 1/4 of the Punnett square. So I'm just kind of trying to hammer home this idea that the area of an outcome square within a Punnett square is proportional to the probability of that combination happening. So we were going to calculate that. We would do it like this, right? And I know you're thinking, yeah, we don't need to write out this math problem because it's so simple right now, but we're going to get into di and trihybrid crosses and it gets complicated real quick, right? So we're trying to figure out the probability of a dominant allele from the mother and a dominant allele from the father, right? This is the probability that we're trying to figure out. So what's the probability of getting a dominant allele from the mother in that cross that you wrote down, right? It's 50%. So 1/2 times the probability of getting the dominant allele from the father, which is also 1/2. right? If we were going to do it, let's say we want to know the probability of getting a dominant allele from the mother. And let's say a recessive allele from the father, right? Father's only got two alleles, a dominant and a recessive. So the probability of this happening is still 1/2, right? And the probability of a dominant allele from the mother is still 1/2. So 1/2 times 1/2 equals 1/4, or 25%. All right? This, and this is big, right? This is not the probability of getting a heterozygous offspring, right? This offspring is heterozygous because it got a dominant allele and a recessive allele, right? So the offspring is heterozygous. But 1/4 is not the process, is not the probability of getting a heterozygous. It's just the probability of getting this combination, the dominant from the mother and the recessive from the father. So, when you're trying to figure out the probability of something happening, all right, the probability of two things happening independent of one another, right? This event, the dominant allele being passed on from the mother, happens independently of this event, right? But in order for this to happen, we need them both to happen. So when two things need to happen together, you multiply their probability together. So if we need this to happen and this to happen, then the probability of them both happening is the probability of this one multiplied by the probability of this one. Right? 1/2 times 1/2. It's just like if I flip a coin, the probability of getting heads and then tails is the probability of flipping a coin twice and getting heads and then tails. It's 1/2, which is the probability of getting heads, multiplied by 1/2, which is the probability of getting tails, right? So I'd have to get heads on the first flip and tails on the second flip. That probability is 1/4, because I need them both to happen. I need the first flip to be heads and the second flip to be tails. So that problem that we just did is actually on your PowerPoint. So if you look at the example problems, the first content slide, it's got 3 problems on there. One of them we just did. What's the probability of an offspring receiving a dominant gamete from the male parent and a recessive allele gamete from the female parent? All right, we actually did it a little different in ours, but the math is all the same. It's the probability of getting the dominant allele from one parent and the recessive allele from the other parent. So we know how to do that. All right, that's fairly straightforward. It's just 1/2 times 1/2. And hopefully you see why now. All right, let's take a look at the practice problems on the PowerPoint. So if you look at the first content slide on the PowerPoint, there's 3 questions. The first question we actually just did. So that was, what's the probability of an offspring receiving a dominant allele gamete from the male parent and a recessive allele gamete from the female parent? That's one we already did. But now let's do the other one. Well, actually, it's the third one on your PowerPoint. It says, what is the probability of an offspring being a heterozygote? Because the reality is that, yeah, that first, the cross, or the outcome in the first question does produce a heterozygous offspring. But that's not the only way that a heterozygous offspring can be produced, right? One possibility to produce a heterozygous offspring is the dominant from the mother and the recessive from the father. The other possibility is the dominant from the father and a recessive from the mother. So to find out, you add the probabilities of all the possible outcomes that lead to that. So we're trying to figure out what the probability of producing a heterozygous offspring is. So one of two things can happen, right? It's the probability of getting the dominant allele from the mother. It's a long day, right? It's the probability of this happening plus the probability of this happening, right? This is one possible outcome. Dominant allele from the mother, recessive allele from the father. That would give you a heterozygote. Here's the other way you can get a heterozygote. Recessive from the mother and dominant from the father. So you add these probabilities together, right? Because either one of them could happen, right? Either this happens and you get a heterozygote, or this happens and you get a heterozygote. So one thing you need to wrap your mind around is when to add the probabilities together and when to multiply them together. So if you would need both things to happen concurrently, then you multiply their probabilities together. If one could happen or the other could happen, then you add their probabilities together. So since either one of these situations would give you a heterozygous offspring, you take the probability of this happening and you add it to the probability of this happening. So we could break this down even further, right? I think that might help. What's the probability of getting that gamete from the mother? All right, I'll let you pause, figure it out. What's the probability of the mother passing on that allele if the mother is heterozygous? It's 1/2, right? Because the mother and the father are both heterozygous. The probability of this happening is 1/2. What's the probability of that happening? Well, it's 1/2, right? Because the father is heterozygous. The father is going to pass on that allele half the time. So the probability of that happening is 1/2. In order for this to happen, you need both of these to happen, right? In order for the mother to pass on that allele and the father to pass on that allele, you need both of these things to happen, right? So you multiply their probabilities together. Now we go over here. What's the probability of the mother, the mother passing on the recessive allele? Again, the mother's heterozygous, so she's going to pass on the recessive allele half of the time. So the probability of that happening is 1/2. The probability of the father passing on the dominant allele, again, is 1/2. In order for this to happen, you need the mother to pass on the recessive allele and the father to pass on the dominant allele, right? Because both of these things need to happen concurrently, in order for this right here to happen, you would multiply their probabilities together. So now, if we're trying to figure out, remember, we're trying to figure out what is the probability of an offspring being a heterozygote. There's two possible ways it could happen. Either this happens or this happens. Either the mother passes on the dominant allele and the father passes on the recessive allele, or the mother passes on the recessive allele and the father passes on the dominant allele. So because either one of these could happen, you add their probabilities together. So if we do this order of operations, 1/2 times 1/2, 1/4, add that to this right here, which is 1/4, that gives us 1/2. So the answer to the question, what's the probability of an offspring being a heterozygote? It's 50%, or 1/2. Here's a good trick to remember this, because you're only going to do two things. There's only two operators that you can use on these numbers. Either you're going to add them together or you're going to multiply them together, right? Those are the only two things. If you need X to happen and Y to happen, if X and Y need to happen, then you multiply them together. All right? If you need X to happen or Y to happen, then you add their probabilities together. So, this isn't contradicting what your second grade teacher told you, right? I know she told you that if you hear and, you add it together, right? Well, that's correct. But not when we're talking about figuring out probabilities. If you need two things to happen, if you need X to happen and Y to happen, then the probability of them both happening is the probability of X multiplied by the probability of Y. So if you see and, you multiply. If you see or, you add. If X could happen or Y could happen, or you add it together. So that might seem confusing to you. And it takes some practice, usually if you're just now starting to wrap your head around this. So we've got a few more practice problems. And it's one of those things, you may struggle with it, but once it clicks, it's just, it turns into this no-brainer. Yeah, of course, this is how it is. Okay. So what about the last problem on that page? It says, what is the probability of an offspring having purple flowers? So this is the probability of having purple flowers from that cross. Right? Well, I don't need to write the cross. You've got it on the PowerPoint. It's a heterozygous cross with a heterozygous. It's probability of getting purple flowers. So, there's a few different ways this could happen. Right? What's one way it could happen? Right? What's one way to get purple flowers from that cross? Well, one way is to get the dominant allele from both parents, right? That would give you a homozygous dominant individual. That individual is going to have purple flowers. So one possibility is that the female passes on the dominant allele and the male also passes on the dominant allele. So we'll have to figure out the probability of that happening. And what's another way that this could happen? Well, another way it could happen is that the mother passes on the dominant allele and the father passes on the recessive allele. So we'll have to figure out the probability of that happening. There's one more way this could happen. And that is if the mother passes on the recessive allele and the father passes on the dominant allele. This would give you a heterozygous individual, and that heterozygous individual would have purple flowers. So we'll have to figure out the probability of that happening. So, to get purple flowers from that cross, either this could happen, or this could happen, or this could happen. Or means that we add them together to figure out the probability of that happening. All right? So, I don't need to break it down into all the many different steps here, but what's the probability of this happening? Right? It's 1/4, because the probability of that happening, right, the probability of a female passing on the dominant allele is 1/2, and the probability of the male passing on the dominant allele is 1/2. So, because we would need both of these to happen together, we multiply their probabilities. That gives us 1/2 times 1/2 equals 1/4. And it's the same here, 1/4. And it's the same here, 1/4. 1/4 plus 1/4 plus 1/4 equals 3/4, or 75%. So what's the probability of an offspring having purple flowers? This is 75%. It's exactly what Gregor Mendel saw, is that when he took his heterozygous F1 plants and mated them with the heterozygous F1 plants, right? You can look back in your notes and look at that. It's towards the beginning. When he took his F1 plants that were heterozygous and mated them with the other F1 plants that were heterozygous, he saw 75% of the offspring had purple flowers. So this is why. So now let's do a dihybrid cross. Everything we've been looking at so far is just a monohybrid cross. So we're going to look at two new characters, with each one with two different alleles. So we're still looking at peat plants. So we're going to look at the character for seed shape, which has two different alleles, and seed color. Pea plants can have two different shapes, or the seeds of the pea plants can have two different shapes. Either they are round or they are wrinkled. The round shape is the dominant allele, so we're going to denote round shape alleles with capital R. And since wrinkled is the recessive allele, we'll denote it with a lowercase r. So this is the dominant allele, and this is the recessive allele. Seed color comes in two varieties, yellow and green. And in the particular strain that Mendel was looking at, the yellow color is the dominant allele. So we'll call it capital Y because that's the dominant. And then for green, we'll do lowercase y because that's the recessive allele. So if we do a dihybrid cross, what we're doing is we're crossing two individuals that are heterozygous for both of these. Or just some combination of both of these. We're looking at two genes, so it's a dihybrid cross. All right, so let's say we've got true breeding dominant plants for both of these alleles. What would be the phenotype if they're true breeding for both of the dominant traits? That means that they are homozygous dominant for both seed color and seed shape. So they're capital Y, capital Y, capital R, capital R. So if you take true breeding dominant and cross it with true breeding recessive, right? These individuals are going to be homozygous for the recessive alleles. So you cross them with them, right? True breeding dominant with true breeding recessive, that's your P generation. What do you get? There's only one thing you can get here. All the offspring are going to be heterozygous. 100% of them are going to be heterozygous in your F1 generation. All right? Now, what if you take one of these F1s and cross them with another F1? It's also from that same generation. So self-pollination. Right? Take the pollen of that flower and use it to fertilize another flower of the same generation. What would you get? Well, let's make a Punnett square. So everybody see what we're crossing? We're going to cross this individual with another individual of the same genotype. So we're going to do this cross right here. All right? That's the cross that we're going to be doing. All right, so each of these individuals, right, they're going to make the same gametes. So before we do our Punnett square, let's look at what the possible gametes are, right? Possible gametes. One possibility is big Y, big R. Another possible gamete is big Y, little r. Another possible gamete is little y, big R. And another possibility is little y, little r. These are the only 4 possible gametes that can be made from either one of these individuals, right? So our Punnett square is going to be 4 by 4 because each individual could contribute one of 4 different gametes. All right? So our Punnett square is going to be quite a bit bigger. There we go. 4 by 4. So we'll put the female gametes right here and the male gametes here. Well, actually, I don't need to write the lines there because we're not dealing with that weird capital P, lowercase p. Right? We don't need the lines anymore. We can just go, here's one possible gamete, right? There's another. Then we got little y, big R, and then the other possibility is little y, little r. These are the 4 possible gametes that can be produced by the female. And the males look exactly the same. Right? Because we're crossing 2 genetically identical individuals. Well, for this gene combination, they're the same. Okay. So, now we start filling in our Punnett square. The individual represented by this box is going to get that gamete from the female and that gamete from the male. So its genotype is going to be big Y, big Y, big R, big R. This individual gets that gamete from the female and that gamete from the male. So again, we're going to be big Y, and then big R, little r. So that's the genotype that comes from that combination. So I'm not going to, walk you through all these combinations. I'm just going to fill them in. So why don't you try this? Why don't you pause the video and you try filling in the rest of this Punnett square, and then when you think you got it, turn it back on and see if you got it right. All right, now pause the video and make sure that your Punnett square looks exactly like my Punnett square. And make sure you understand why everything is arranged the way it is. All right. So, by now your Punnett square should look just like my Punnett square. And you should remember that each one of these squares represents an equally likely event, right? This square represents the event where that gamete came from the female and that gamete came from the male. And so on and so on and so on. So what are, let's see here. Yeah. What are possible phenotypes? right? Well, one possibility is that we had yellow round seeds, right? Yellow round seeds. That's one possibility. I'm going to color code these for you. Well, you know, we can go ahead and we can circle all of the individuals here that would have that phenotype. So pause the video and try and find all the individuals that have the yellow round phenotype. And I'll do it. All right, did you guess right? Hopefully you didn't guess. That one's obviously yellow and round because it's homozygous dominant for both traits. This one's yellow because, again, it's homozygous dominant, but it's heterozygous for the round gene. But since round is dominant, round is what gets expressed. So it's yellow round, yellow round, yellow round. All the ones that I circled in red are yellow round. That's one possible outcome from the cross. Another possible outcome is that we got yellow wrinkled seeds, right? Wrinkled is the recessive allele for the seed shape gene. So we'll go through again, pause the video, try to find all the individuals that have yellow wrinkled seeds. And I'll circle them. That's it. There's only 3 of them. Another possibility is that the seeds are green and round. And it's unfortunate my brown marker looks a whole lot like my red marker. Okay, so we'll circle, again, pause the video, and try and find all the individuals that have green round seeds. There they are. And the last possibility is green wrinkled seeds. There's only one way that can happen. That right there. So, the ratio that you see in a dihybrid cross, where both individuals are heterozygous for the 2 genes that you're looking at, right? That's a heterozygous cross. The 4 outcomes that you see, dominant, 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 recessive, recessive, dominant, and recessive, recessive, happen at a fixed ratio. 1, 2, 3, 4, 5, 6, 7, 8, 9. For the green ones, 1, 2, 3. For the brown ones, 1, 2, 3. And for the double recessive, 1. So you see these 4 outcomes happen at a 9 to 3 to 3 to 1 ratio. All right. Let's do more math. All right, now it's getting a little bit more complicated with the dihybrid cross. So if you look at the next slide on the PowerPoint, let's do that first question. What is the probability of a female YR gamete uniting with a male, oh, I'm sorry, not a YR. It's all YR. All right, what's the probability of an offspring receiving a double dominant gamete from the male parent and a double recessive gamete from the female parent? That's just the first question on that next PowerPoint slide. Probability of getting an offspring that receives that one from the male parent and then a double recessive gamete from the female parent. Well, you could just draw a Punnett square, right? All you got to do is find the squares that represent that event happening, and there's only 1, 2, 3, 4 times 4. There's only 16 squares here, so it's, you know, the number of squares that happens in divided by 16. So you could just say, okay, well, a double dominant gamete from the male parent, where's that? Okay, right here. And a double recessive gamete from the female parent. It's right there. Okay. Well, there's only one square where that happens. So you could just draw out a Punnett square and say, okay, it's 1/16. That's the probability of it happening, 1 time out of 16 times. But you don't want to have to draw a Punnett square every time you do that, because that's excessive. And you don't need to. Let's rewrite the problem so you can see how to just do it in your head really quick. All right? So it's the probability of a double dominant from the male parent and a double recessive gamete from the female parent. All right? It's this has to happen, and this has to happen. So what do you do with these two probabilities? You multiply them together. So what's the probability of getting that gamete from the father? Right? It's 1/4, because each individual can only make 4 different gametes. They're all unique, right? So the probability of getting any one of those 4 is 1/4, right? So it's 1/4 and times the probability of getting a double recessive gamete from the female. Again, female can only make 4 different gametes for this particular cross. And the probability of getting any one of those 4 is 1/4. So it's 1/4 times 1/4. 4 times 4 is 16. 1/16. There you go. That's the probability of getting that specific combination of gametes. That's pretty simple. The next one's a little bit trickier. What's the probability of getting this genotype? All right, that's the second problem. What's the probability of a double heterozygous genotype from that same cross. All right? So you got to think of all the different ways that this could happen. One possibility is that, let's see here. I'm going to do this. Make sure I got enough room to do this. All right. One possibility is that you got a big Y, big R from the female, and then what would you get from the male in this case? Well, you would get a little y, little r. That's one combination that will give you that genotype. All right? That's one way that it could happen. Or something else could happen, right? So, or means you add the probability of, well, I'll just put plus, since we know that or means add. So we add to the probability of this happening the probability of everything else happening that could lead to that genotype. Right? Another way this could happen is the female gives a double recessive gamete and the male gives a double dominant gamete. Right? That combination will also give you the double heterozygote. What's another way it could happen? Well, there's also the probability that the female gives that gamete and the male gives that gamete. And there's one more way that this could happen, and that is if the female gives, what was the last way? Yes. If the female gives that one and the male gives that one. So, yeah. These are all the different ways that you could get this phenotype. There's a certain probability that this could happen, right? There's a certain probability that this could happen. There's a certain probability that this could happen. And there's a certain probability that this could happen. Any one of them could give you that genotype. So it's this, or this, or this, or this. And since any one of them could happen, we just add their probabilities together. So the probability of this combination happening, that's what we figured out on the last problem, right? The first problem on this PowerPoint page. The probability of this happening is 1/16. And you would just add to it the probability of this happening, which is 1/16. This is also 1/16. And this is also 1/16. You add them all together, you get 4/16, which is just 1/4. So this is the probability of a genotype that is heterozygous for both of those genes. And you can just count them on the Punnett square, right? You probably still have the Punnett square written in your notes. And, you know, we're still dealing with that same cross. So go back to that Punnett square and count all the individuals that have that genotype, and you'll find out that it's 1/4 of them have that genotype, right? 4 out of the 16 squares in that Punnett square have that genotype. All right, and the third question. What is the probability of a yellow wrinkled phenotype? Again, just think about all the different ways that it can happen. All right? So, yeah, we're looking at the third question here. What's the probability of this phenotype? Yellow, wrinkled. Well, it's just the probability. There's only two genotypes which can give you this. So let's break it down that way, right? It's the probability of that genotype, right? That would give you this phenotype. Big Y, little r. That'll give you big Y, little r phenotype. All right? There's another way. I'm going to spread this out quite a bit, actually. Well, right there. So this could happen, or this could happen, right? So these are the only two ways that you could get that phenotype. This genotype and this genotype. That's the only two combinations that can give you this particular phenotype. All right? So we just need to figure out what the probability of this is and then what the probability of this is. So what's the probability of that? Well, there's really only one way that could happen, right? To get that genotype, you would have to get a big Y, little r from the male, and a big Y, little r from the female. So, let's see here. I'm actually going to color code this just so you can see it. So it's probability of big Y, little r. All right? So, there's only one way this can happen. So this is equal to the probability of the female giving big Y, little r, and the male giving big Y, little r. Right? Everybody see that? Because that's the only way that this could happen. Right? There's no other way it could happen. What about this one? Well, you could get a big Y, little r from the male and a little y, little r from the female, or you could get a big Y, little r from the female and a little y, little r from the male. So there's two different ways this could happen. So we'll expand this just a little bit and say it's the probability of the female donating big Y, little r, right? And the male donating little y, little r, right? That's one way it could happen. Or the female donates a little y, little r, and the male donates a big Y, little r. That's the other way this could happen. Right? And I'm going to break it down even further. You can stop the simplification whenever you need, but I'm going to break this down even further until we just get down to the 16ths, or the 1/4 chances actually. So what's the chance? Let's look at this right here, right? What's the probability of this happening? Right? There's two things that have to happen in order for this right here to happen. The female has to donate that gamete, and the male has to donate that gamete, right? So the probability of this happening is the probability of that happening times the probability of that happening. So the probability of the female donating that gamete, it's only 4 gametes, and it's random, so the probability of it happening is 1/4. So for this, we need to know the probability of this happening and that happening. So what do we do with those two probabilities? We multiply them together. 1/4 times 1/4, right? That is the probability of this happening. Oh, we can do the same thing over here. What's the probability of the female donating that gamete? Well, the female's only got 4 gametes, and the probability of any one of them is 1/4. So again, it's 1/4 times the probability of this happening, which is 1/4. Right? We can add those together. And the same thing over here. It's 1/4 times 1/4. Right? So does everybody see how we got that? So 1/4 times 1/4 is 1/16. This is 1/16. And this is 1/16. So the probability of getting this phenotype is 1/16. I'm sorry, is 3/16. And again, you can count them in the square, right? When we did that 16 square, Punnett square, and you circled. You did this earlier. You circled all of the big Y, little r phenotypes, right? And you probably noticed that there were 3 of them there, right? So that 3 out of 16 represents the probability that you get that phenotype. All right, so let's look at the last problem on that PowerPoint slide. And it looks like, a hard one because, you would need to, if you're going to do this with a Punnett square, your Punnett square would need to be, way too big. It would take you half the class period just to draw out the Punnett square and get it all filled in. So this is where you need to be able to do it mathematically. But we'll break it down into the simplest steps that we can, and you'll see it's really not as hard as it looks. So for this particular problem, all right, here's the cross we're doing. We're bringing back our flower color gene, and we're going to cross an individual with this genotype with an individual with this genotype. And what we want to know is the probability of getting this phenotype, right? So, for this to occur, 3 things need to happen concurrently. We need a recessive seed color genotype, or we need a recessive seed color phenotype, a dominant seed shape phenotype, and a dominant flower color phenotype. So we need all 3 of these phenotypes to be expressed at the same time. So we just need to figure out the probability of each one of them and multiply them together, because we need this to happen, and we need this to happen, and we need this to happen. Make sense? So we'll do it like this. First of all, we want to know the probability of a little y phenotype, right? So we need that to happen, and we need a big R phenotype. So it's the probability of the big R phenotype. And we need the purple flower phenotype, right? So we need this to happen, and we need this to happen, and we need this to happen. And these are a lot simpler to solve than the whole thing together. So let's start simplifying. Well, how many different ways could this possibly happen, right? There's only one way this could happen. You get the recessive allele from this individual, and you get the recessive allele from this individual. So it's the probability of a homozygous recessive genotype, right? And we're going to multiply that by the probability of this happening. So there's actually two ways that this could happen. Either we get the big R from here and the big R from here, or we get the big R from here and we get the little r from here. Those are the only two ways that will give us a big R phenotype. So either we get the big R from here and the big R from here, so that probability, or we get the big R from here and the little r from here. Right? And these are much more manageable problems, easy to solve. So we'll take that probability and multiply it by the probability that we get a purple flower phenotype. Right? And there's only one way this can happen. The only way that we can get purple flowers from this cross is if we get the big P from here and a little p from here, because we're always going to get a little p from here. So it's the probability of this genotype. All right, this is simple enough. So we'll simplify it even further. And once you get this, you can fast forward, right? You don't have to simplify it in as many steps as I'm doing. I'm just breaking it down for the people who are trying to wrap their heads around it. So, in order for this genotype to occur, two things have to happen. You have to get the little y from here, and you have to get the little y from here. So you need that y to be donated, and you need that y to be donated. The probability of getting that y is 1/2. You need this to happen. You get that y and that y. So you multiply their probabilities together because you need them both to happen. So it's 1/2 times 1/2. That's the probability of getting a homozygous recessive genotype for that gene from this cross. 1/2 times 1/2. Right? And you could figure that out with, you know, single gene Punnett square. All right. What about this one? Well, what's the probability of getting this genotype? How many different ways could that happen? You would have to get the dominant allele from here and the dominant allele from here. All right? That's the only way this could happen. So what's the probability of getting the dominant allele from here? 1. So it's 1 times the probability of getting this dominant allele, which is 1/2. All right, so we'll add that to the probability of getting this genotype, which is the probability of getting, again, that allele, which is 1. Multiply that by the probability of getting this allele from here, which is 1/2. So we've simplified it even more. Multiply this whole thing by the probability of getting this genotype, which is going to happen, again, half the time, because the probability of getting the small p from here is 1. So it's 1 times the probability of getting the big P from here, from here, which is 1/2. All right, and there we are. You just have to add all this stuff together, and then that'll give you your answer. So, 1/2 times 1/2 is 1/4 times 1 times 1/2 is 1/2 plus 1/2 times 1/2, right? So this is just 1/4 times 1, right? 1/2 plus 1/2 is 1, times 1/2. So 1/4 times 1 times 1/2. All right? And so it's just 1/8. That is the probability of getting a little y, big R, big P phenotype. It is 1/8. And if you wanted to, you could draw out the whole Punnett square. It would be 8 by 8. And so it would take you a while to fill it in. And that's even more tedious than what we just did, I think. So it's never actually that easy. These examples that we were looking at so far, everything we've been looking at is an example of what is called complete dominance. And with complete dominance, the dominant allele is always expressed over the recessive allele. And if you have a heterozygote, then the recessive allele is completely masked and you don't see it at all in the phenotype. This is the exception. It's not the rule. Most genes don't work like that. So before we go into the other ways that it can happen, we'll refine our definition of recessive. So what makes an allele recessive is the fact that it can be masked by a dominant allele. All right? So a recessive allele is recessive if it can be masked by a dominant allele. So this is true for all the other different modes of inheritance. So we'll look at another one. All right, incomplete dominance. So with incomplete dominance, heterozygotes actually express both alleles. So let's say you have one gene or one allele that can give you red flowers. And then another allele that can give you white flowers. This is in a different plant. Snapdragons, not peas. We're not talking about peas anymore. We're talking about snapdragons. It's another flower. I should put a picture of snapdragons in my PowerPoint, but I don't have one. You look up a picture of snapdragons. Yeah, so both of these alleles are dominant, but they have incomplete dominance. So both of them get expressed. So there's three possible genotypes here. One is that, one is that, and the other is that. So a plant with that genotype is going to have red flowers, and a plant with that genotype is going to have white flowers. And a plant with that genotype is going to express both of these alleles, so it's going to have pink flowers. So a dominant allele is not dominant if it can mask another allele. It's dominant if it cannot be masked, right? You rewind that and listen to that again. So that's incomplete dominance. Another mode of inheritance is codominance. In codominance, you can have two different alleles for the same gene that are both expressed in different ways. So in codominance, both alleles affect the phenotype in different ways. And a really good example of this is blood type. So we talked about blood type earlier on in the semester. And so now we're going to look at how your genes actually determine blood type. So for the blood type gene, there are three different alleles. All right? There's the I superscript A allele, the I superscript B allele, and then there's the lowercase I allele. Right? These are both codominant. Right? This is recessive. So this is the recessive allele. And these are codominant alleles. So if you remember back, actually, you know what I would do? I would hit pause on this, and I would go look at my notes from, I can't even remember what chapter this was. I think it was the cell chapter. I think it was chapter 5 or 6. I don't remember. Go back and look at the cell chapter and look at blood type, because the thing that determines blood type is the carbohydrate that's expressed on the outside of your red blood cells. So go do that and come back. And now you're back. All right. So this allele encodes for the A sugar on the outside of the cell. So the carbohydrate expressed by that allele is the A carbohydrate. And the carbohydrate expressed by that allele is the B carbohydrate. This allele doesn't encode any carbohydrates. Right? So, what are the possible genotypes? You could have that one. 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 What else? You could have that one. Or you could have the double recessive. Is that it? I think that's it. That's it. All right, so these are the 6 different possible combinations of these 3 alleles, right? So the carbohydrates that are expressed by this genotype is just A. Right? For this one, this recessive allele is masked by that dominant allele, so we get the A carbohydrate. This one and this one give you the B carbohydrate. This one and this one also give you the B carbohydrate because that recessive allele is masked. But here we have 2 codominant alleles. So they're both going to be expressed in different ways. The A allele is going to cause the expression of the A carbohydrate, and the B allele is going to cause the expression of the B carbohydrate. So that's why you get type AB blood, because both of these carbohydrates are being expressed. With this genotype, you get 2 recessive alleles, neither of which encode a carbohydrate, so you get nothing. That gives you type O blood. So this will give you type A blood, type A blood, type B blood, type B blood. This will give you type AB blood, and this will give you type O. So this is just one example where you can have 2 alleles for the same, more than 2 alleles for the same gene. It's not always as pretty as the pea plants that we've been looking at. You can have 3, 4, 5, 6 alleles for the same gene. Sometimes one gene can affect multiple different characters, right? In that case, you're looking at a thing called pleiotropy. This is when a gene has multiple phenotypic effects. Right? This is when one gene has multiple phenotypic effects. Right? That's pleiotropy. You can also have polygenetic inheritance. This is when more than one gene controls a single phenotype. Right? I'd write that down. I'm not going to write it down. You write it down. It's when more than one gene controls a single phenotype. A really good example of this on the next slide is skin color in humans. Right? This example is on page 282 in your textbook. And this is, pretty oversimplified, but it's a really good example of how more than one gene can control the single phenotype of skin color. This is why you get all different shades of humans. You can have people that are very dark skinned and people that are, very light skinned. So in this case, there's 3 genes that are controlling the phenotype of skin color in humans. And so you can see how all the different combinations give you different shades of skin color. Another mode of inheritance called epistasis. All right? This is when the expression of phenotype of one gene alters that of another gene. And you can go ahead and go to the next slide. Aww. Yeah, Labrador puppies. This is a perfect example of epistasis. So, in Labradors, there's one gene that controls the color of pigment in a hair. And then there's another gene that controls whether or not that pigment actually gets deposited in the hair. So, in Labradors, there are 2 genes that control coat color. All right? You've got one that determines the actual color, right? And there's just, regular old complete dominance here. Either it's black, which is the dominant, or the color is brown, right? So that's what determines the pigment, right? Either the pigment is black or brown. And this is just a regular old complete dominance right here. But there's an epistatic gene. That determines whether or not that pigment actually gets deposited in the hair. And there are 2 alleles for that. There's the pigmented allele, all right, that's the dominant one. And then there's the non-pigmented. which is recessive. Right? So what would be the phenotype of an individual that looks like this? What we're doing here, yeah. All right. Well, the pigment color is black, right? Because it's homozygous dominant. And that pigment does get deposited, right? Because it's got the dominant allele here. So this individual would be a black lab. A brown lab might look something like this. All right? It's got the brown phenotype, right? Homozygous recessive. It's going to be brown, and that pigment is deposited, so you get a brown lab. But a white lab only happens if you get a homozygous recessive genotype for the epistatic gene. So if you get anything with the homozygous recessive there, right, whether you're like this or like this or like this, any one of these is going to give you a white lab because that pigment is not going to be deposited. So you can look at that, you got 4 litters of puppies there. And you might be able to figure out what the genotype of the parents were for each litter of puppies just by looking at the phenotypic ratio of their offspring. Which I'm not going to do right here, but you can try to figure out and come to my office hours and see if you got it right. It's a good exercise for you. All right. All right, so I'll talk about a few different genetic disorders. Well, I want to talk about one, just as an example of a genetic disorder. Sickle cell disease. This is a genetic disorder that is caused by a recessive allele. So in order to get this disease, you would have to have 2 of the recessive alleles that encode a protein called hemoglobin. So you can jump ahead next slide. On the left side, what you're looking at is normal hemoglobin. And on the right side, you're looking at the sickle cell hemoglobin. And there's just, there's one little mutation in that gene that encodes the recessive sickle cell disease allele. And what happens is if you've got this, if you're homozygous for the recessive version of this gene, then the hemoglobin, which is a protein in your red blood cells that carries oxygen, you can jump ahead to the next slide. The hemoglobin protein will chain together because of this one little mutation. It'll chain together, and you get these really long chains of hemoglobin that can stretch out that red blood cell and make it long and pointy, like you see in that picture right there. And so the cells will actually take on the shape of a sickle, and that's why they're called sickle cell disease. You can come back to the video. You can't do a test cross with humans, right? That's not exactly ethical, right? So if you want to find out the genotype of humans without doing a test cross and without sequencing their DNA, I know it's a lot easier these days, right? What you can do is you can look at the history of genetic disease in somebody's family, and a lot of times you'll be able to figure out the genotype of an individual even if they're not expressing that recessive allele. So you do this with a pedigree. And pedigree is just a history of trait inheritance. It's like a family tree that tracks the inheritance of a particular gene. So when you're drawing a pedigree, we'll draw the males as squares and the females as circles. So a square that looks like that is a male, and a square that looks like this, all right, filled in. This is how you indicate a male with the particular trait that you're looking at. And you do the same thing with females. This is how you indicate a female with whatever trait it is that you're looking at. All right, so here's an example. We'll do it like this. Here's an example pedigree of, we'll say, sickle cell disease. So you have a male with the trait mating with a female with the trait, and they have, we'll say, 4 offspring. 1 that is an affected female, 1, and I'm going to draw it down low like this, and you'll see why in just a minute. 1 that is an affected female, another that is an affected male, and then another affected female. All right, so this affected male mated with an affected female, and all of their offspring had whatever trait it is we're looking at, in this case, sickle cell disease. And this female here manages to find herself a beau that doesn't have sickle cell disease. And so they mate. And they have 4 children. A male that's not affected, another male that's not affected, a female that is affected, and a female that is not affected. All right? So, you could analyze each individual in this pedigree, and for a lot of them, you'll be able to, actually for all of them, you'd be able to figure out their genotype. If this is a recessive trait, right? The trait that we're looking at for sickle cell disease is recessive. We can figure out the genotype of every one of these, right? Well, right off the bat, you know that if they express that recessive allele, they are homozygous recessive, right? Homozygous recessive, homozygous recessive, homozygous recessive. All these dark ones are homozygous recessive. This one right here got the recessive allele from this parent, and it had to have gotten the recessive allele from this parent, right? So we can look at this one right here and determine that they must be heterozygous. because they're not expressing the recessive allele, but they had an offspring that is. So this individual had to get the recessive allele from this parent. So we know that this individual is what we call a carrier, right? So another way to denote this is like that. Fill in half like that. So because we saw that, we know that this individual is a carrier, right? And we can look at these other three individuals and we can tell their phenotype as well, because we know that each one of these offspring got the recessive allele from this parent. Had to, right? Because this parent only has one allele to give, and that is the recessive allele. But because they don't express that recessive phenotype, we know that they must have gotten the dominant allele from the father. So we know that they're all carriers. It's an unfortunate family tree, right? So, yeah, carriers. There's a word for you. These are just individuals that are heterozygous for a recessive disorder. And on the very last slide, got a practice problem for you there. You can pause the video and read it and see if you can figure out how to tackle that problem. So, let's figure out Jim's genotype, right? Jim's genotype has got to be, let's say, let's see, Jim. Okay, well, let's look at sickle cell disease first, right? Let's do Jane first. That's a little bit easier. All right, Jane is a healthy woman whose father has sickle cell disease, which is a recessive disorder. Okay, well, if her father had sickle cell disease, which is a recessive disorder, he had to have been homozygous recessive, right? So no matter what, she's going to have that recessive gene. But she's healthy, so we know that she's got to be heterozygous. So there's her genotype for sickle cell disease. What's Jim's genotype? Jim has no family history of sickle cell disease, right? Far back as he knows, nobody in his family has ever had sickle cell disease, so he's got to be, he's true breeding non-sickle cell, so he's homozygous dominant for the sickle cell disease. All right, what's the next one? We'll do achondroplasia. So both Jim and Jane have achondroplasia, right? Which is a form of dwarfism caused by a dominant allele. And both Jim and Jane have one dwarf parent and one normal parent. So we know that the normal parent had to have given them the recessive allele for achondroplasia. So, because they each have a normal parent, they each have at least one copy of that recessive allele. But since they both have achondroplasia, we know that they also have the dominant allele. So both of them are heterozygous for the achondroplasia gene. All right? Huntington's disease. Jim has Huntington's disease. That's unfortunate. Which is a disorder caused by a dominant allele. His mother also had Huntington's, but his father did not. So he must have gotten that dominant allele from his mother. And his father did not have Huntington's, so his father was homozygous recessive. So Jim must be, oops, Jim's down here. Jim must be heterozygous for the Huntington gene. What about Jane? Well, Jane doesn't have Huntington's disease. So she's going to be little h, little h. All right, well, now we were just able to do some detective work and figure out the genotype for each one of these individuals. So, what's the problem? If Jim and Jane mate to produce one offspring, what is the chance that their offspring is normal for all three disorders? All right, so you pause and try and figure this one out. It's the probability of not having sickle cell disease. So, what's the probability of them having sickle cell disease? Or what's the probability of them not having sickle cell disease? It's actually 1, because Jim is always going to donate his non-sickle cell gene, or his non-sickle cell allele. So no matter what, their offspring is not going to have sickle cell. Well, that's nice. So it's, well, we'll just do it like this. 1 times the probability that they have a normal non-dwarf offspring, which is caused by the dominant allele. So, well, it's actually 1/4. Because it's the probability of getting that allele times the probability of getting that allele, right? That's what would have to happen if they want to have a non-achondroplasic offspring. Have to get that and that. So it's 1/2 times 1/2, which is 1/4. All right. And then the probability of the offspring not having Huntington's disease, which is really, it just depends on whether or not Jim donates this allele or that allele. If he donates this allele, then the individual has Huntington's. If he donates this one, then the individual does not. So there's a 50-50 chance. All right, 1 times 1/4 times 1/2. They have a 1 in 8 chance of producing an offspring that does not have any of those genetic disorders. So yeah, so that's the end of chapter 14. There's no chapter 14 quiz, and I think you'll see why after we do chapter 15, because chapter 14 is just, it's kind of an incomplete picture as far as inheritance goes. Once we get done with chapter 15, you'll have a lot better idea at how this actually all works. So the chapter 14 quiz is going to be lumped in with the chapter 15 quiz.